materials for promotion to full professor. Matt?
All right, well, thanks, and thanks y'all for showing up. I know it's hard to get more than 5 of us in a room at any one particular time and place anyway, so I've got some slides. Hopefully it won't take that long, but again, last thing I wanted to do was essentially just sort of read you the dossier. I mean, it's sort of dry as it is in terms of going through all the old stuff, so trying to turn this thing into some sort of a story. So I'm sort of, again, sort of following a template I used the last one of these. For the most part, everything that I'm working on sort of falls under this umbrella of looking at immunity and resistance to disease, or ways we can prevent disease in poultry production, be it turkeys or chickens, whatever else. The top 3, and these things are all equal size, but they might not necessarily be equal representation at a particular time or place on things we got going on, but the research stuff on the top, and then teaching, mentoring, outreach service type stuff on the bottom. Sort of go through all the stuff sort of blends together, all of them touch on immunity or disease. And so I'm just going to tell you a little bit, some of the stuff, each one of these things in kind of sort of highlights over the last several years of stuff that sort of to pull out of the package that I think have been some of my major contributions to poultry science, and maybe even to hopefully the science in general. So sort of starting off with the microbiome, one of the newer projects and major focuses of the group right now. A lot of this you guys see presented to the department here recently, or Anne presented her thesis, and some of the stuff you've seen before. So I'll go through it fairly quickly, but just for background purposes, we've been with the microbiome, for the most part, what we started off with years ago was looking at how probiotics, specifically Premolac, affected the gut health and immunity of mostly in broilers. We've done some stuff in laying chicks as well. So with this, initially we found that feeding the Premolac affected gut development and made histological changes. Here I'm showing you data from the ileum, but we've seen it all up and down the GI tract. Again, this isn't meant to be a sort of data presentation, so I'm going to go through these fairly quickly. But if folks have questions, feel free to stop me. I can geek out on this stuff for hours. But the changes histological following probiotic treatment isn't necessarily new or novel. Lots of folks have reported these types of things. Where we start to see differences in our model from what others have reported is when we start looking at how the animal, if you get bigger tissue, if you get longer villi, but the birds are growing just as well, what does that mean in terms of how energy is being redistributed throughout the body? And what we found by looking at either the whole body, there was really no difference between treated and control in terms of energy metabolism and respiration rates. The intestine actually seemed to, the ileum specifically, be decreased in energy consumption, where what I was most interested in and what we sort of been focused on is increased energy consumption by immune tissues. And this is sort of summarizing 2 PhD students' worth of work. So the table here sort of shows you transient. You see the ileum's down day 15 and day 22. The thymus is up at 8 and 15. They're not necessarily 1-for-1, but the body's using energy in different locations than it is in the controls. So from there, we wanted to know, okay, well, this immune tissue that's actually being affected by probiotic treatment, can we look at immune cells in circulation? So we developed an assay where we can actually isolate, and here we focus on pretty much all the white blood cells, PBMCs, not just lymphocytes. That'll change in a couple slides. But, so we see any evidence that the immune cells themselves were behaving differently or had different energy statuses. So by isolating these cells, and then we sort of repurposed another assay that uses luciferase and the ATP liberated by cells as a way to quantify ATP per cell. So we go through, we count the cells, we isolate them from controls for treatment, put them on this ATP luciferase assay, and what you see is more ATP per cell in probiotic cells isolated from probiotic fed birds versus controls. And then actually when we do incubation studies, we can see that the probiotic fed animals, actually while they start off with more ATP, they're burning through it faster. And so it's indication, or at least suggestive, that they're at sort of a higher energy status. This higher energy status also correlates, at least time-wise, with when we see a faster antibody switching, or antibody isotype class switching. So we immunize the animals and this stuff because we're doing energy stuff. We didn't do an actual infection. We did a sheep red blood cell challenge just to sort of give an antigenic challenge to give the immune system something to get mad at. And what you see is IgM, which is sort of the default pathway for all immune responses. It's about the same between treated and control. But IgG shows up 10 to 14 days sooner in the probiotic-fed animals versus the controls. And that's antigen specific in the serum, and then when we do gut washes, we actually see higher levels of IgA total. We don't get antigen specific responses in the gut, but we get higher total IgA in the probiotic-fed groups versus the control. So again, evidence that something about what the probiotic is doing to the immune system is driving some sort of class switch and stimulating a faster response of some sort. So what we really want to do is try and figure out what that is. And so on a lark, one student decided to do something that I told him would never work, and it turns out it did, where we actually bled the birds and take serum from probiotic-fed animals or serum from control animals, put that on lymphocytes in culture, and then after 48 to 72 hours, come back and measure how much ATP is there. And so here's 4 different cell types. The CU205 is the T cell-like cell line. RP9 is the B cell-like cell line. This is a liver cell, and this is a macrophage cell. And essentially, after 3 days of incubation with serum from probiotic-fed animals, you get higher ATP per cell in T cells and B cell-like lines, but not in these other non-lymphoid-like or non-lymphocyte lines. So there's something in the serum that is communicating this go or no-go signal to immune cells, driving some sort of response. We don't yet know what that is. We've been doing sort of classical biochemical size fractionation, delipidation. Is it a protein? Is it a metabolite? Is it a lipid? It seems that it may be a lipoprotein somewhere in the 15 to 30 kilodalton range, but we haven't been able to narrow it down any tighter than that. And the folks in the mass spec lab over in chemistry don't think we've got the prep clean enough that they want to put it on their magnet. So we've now been working on ways to sort of back into it. We've taken these cells, the T cell lines, because we get the biggest response or fold difference there. Taken those and done RNA-seq, trying to figure out what is the compound, what genes are to turn on or turn off, and can we use that as a way to back into what's the ligand, trying to figure out what is the probiotic actually stimulating. But for the most part, the model we've been building, and now what we're working towards, is as we feed probiotic in this end, and we change the gut architecture and the microbiome all along the GI, which is what these panels supposed to represent, that we've got shifts in the biome, that we're getting changes in metabolite production. And right now, what the working hypothesis is that as we change the taxa, or we're at least stimulating different gene expression in the taxa that are there, we're getting some sort of metabolite that's probably passively diffusing across the gut barrier or stimulating the gut to produce some other factor, who knows. But that's then talking to the immune pathway, but then that's what's leading to enhanced antibody production or whatever immune function. So this is sort of what we're continuing to work towards is, we can show the taxa is different, we can show we get antibody differences, we know there's stuff in the serum, and we'll get something, somehow, some way being communicated across the gut. So now most of the efforts in the lab are starting to focus on, can we identify this signaling molecule, whether it's a metabolite or otherwise, that's actually the bridge between these two stages. So again, sort of tearing through a lot of different projects fairly quickly. So still in the microbiome, one of the other sort of prong to the microbiome project has been looking at how does changes in the microbiome affect resistance, specifically to salmonella. And here, this is some data where we start off with Dale, these are actually pullets, and we gave them a live attenuated salmonella vaccine, a Typhimurium vaccine at day of hatch, and then fed them either probiotics or control diets, and looked at how does the addition of the probiotic with and without the salmonella change the microbiome, who wins in that sort of turf battle as all these bacteria are fighting for resources and for space, and then sort of follow from day 1, day 7, all the way up to 28 days, sort of follow the development of the microbiome. And for the most part, the biggest take-home message is, even though salmonella gets in there early and is gone within 14 days, the fact it was there, it sets up the biome on a path that continuously feeding the control or the probiotic diet by itself never really brings it back, and by 28 days, you have 4 distinct different biomes. And so, there's nothing wrong with these birds. These are just 4 different ways that a healthy biome can be there. So, just because we can change it doesn't mean we change it in a good direction or a bad direction. Sometimes the difference is just different. But still trying to go through, there's so many papers that every time they change the biome, they try and otherwise make it sound like they've cured cancer or invented a longer lasting light bulb. But sometimes it's just different. So now that we've got 4 different types of what seem to be normal, we're trying now to tease out who's always there when the bird's healthy, who's not there when birds may be more prone to colonization by salmonella and other stuff. And so when we start to actually identify more of a phenotypic microbial population that's better correlated with health or some sort of status other than just different. So if this was done with probiotics, this is still the Premolac and attenuated salmonella vaccine strain. So we got other projects. This one's part of the collaboration with OSNI. We're actually looking at prebiotics, and can we, instead of adding the bugs that we think that should be there, whether they're the right bugs that should be there or not, can we add the substrates that the right bugs are already there and otherwise encourage them to expand and take over as opposed to having to keep feeding live organisms. And also, here we're using actually a cocktail of wild-type salmonellas, Typhimurium and Enteritidis. But sort of similar design where birds got a control diet, they were challenged with salmonella orally, or they were given a prebiotic with the salmonella challenge, and then follow the development of the biome. And here again, the control, everybody ends up being different from control. After about by 27 days, 28 days, the control plus salmonella, prebiotic, and prebiotic salmonella are all more or less, they're not the same microbiome, but they've started to drift towards each other, and the control stays off by itself. Again, there's nothing wrong with these birds. They're all relatively healthy. And the prebiotic does sort of seem, it's transient because the Enteritidis goes away pretty quickly on its own. Typhimurium is on its way out by 28 days, and as we go a little further, levels go down. There's always maybe a couple of positive birds for a few more weeks in some experiments, but not always. But the prebiotic seems to be helping hasten the exclusion of salmonella. I'm not really sure what word I'm looking for there. And so this has given us at least enough of a foothold to come back and try, okay, well, what bugs are the prebiotics encouraging? Can we add those? So we've done experiments where we've added the prebiotic plus taken cecal samples, or Koci taken cecal samples, and cultured those with the prebiotic in the lab and selected for organisms that can grow and can metabolize the prebiotic, and then add the prebiotic as well as those organisms as sort of a prebiotic-probiotic cocktail, and looking at different combinations, ways that we can sort of drag those two apart from each other and enhance the salmonella going away.
Can I ask you something?
Please.
Go back. So looking at this, only in those 2 points is that they are different.
Correct.
So the prebiotic doesn't match Salmonella typhimurium or the Enteritidis.
So for the most part, at least in our hands, with the strains that we're using, the birds don't care. And so I think to some extent, what I'm not showing, this is cecal data. We've got liver and we got spleen data as well. In both cases, Enteritidis almost never gets across the gut. Typhimurium does, but it's gone within a couple of time points. In some experiments within 14 days, in some experiments maybe within 5 days. it'll get across, but I think as soon as it gets across and it's in the cecum and it's not really causing the bird any problems, then there's really no reason for the bird to expend any more energy to get rid of it. Other strains of salmonella that may be a little bit more bird aggressive, or more likely to want to invade a chicken more so than some of these strains that are more. I mean, these are all strains that were isolated from humans but associated with poultry products that got them sick. And so, how to tell those two apart? But yeah, no. To your question, right now the prebiotic does a little bit, maybe early. I think this one, it goes away so fast, it's really hard to make a big difference. Here, it's transient at best. Prebiotic's not doing much. But this, while unimpressive, at least initially, it's been a consistent across a couple of experiments. So there's enough going on there that was, okay, maybe it's a different dose, maybe more probiotic, or a prebiotic, less prebiotic, putting the bugs in. That's sort of why we're trying the prebiotic and the probiotic combo. Can we turbocharge this a little bit more and draw those two apart?
Also, these birds were infected areas.
No, yeah, so that's another good point too. So the data from this, these birds were all infected essentially around day 3, day 4. When we do the same experiment where the birds don't get infected until 4 weeks of age, you almost get no colonization. So there's this, and it seems to be an interplay, and we're trying to go back and figure out ways to do this experimentally. When we've done one experiment with SPF birds that have no maternal antibodies, colonization happens a little bit better, still not fantastic, the birds don't get sick, but there sort of seems to be this window that with commercial birds, we're all doing all these with commercial highline birds, trying to make this as real world relevant as possible. Mom's antibodies are through the roof. Maternal antibodies seem to protect the birds at least to invasion for the first 3-ish weeks. And it takes about 3 to 4 weeks for the biome to establish. So it's almost like mom's antibodies keep salmonella in check until the microbiome gets established. If you challenge after the mom's antibodies are gone, but the biome's established, salmonella has no shot. The only way you really get a good, this isn't really good, the only way you get somewhat sustainable salmonella colonization is to infect early while the biome is still establishing, and salmonella can set up a niche. Once it's a little bit older, then it's a lot harder to get salmonella to hang out. So, I think that's age, clearly it's microbiome development, mom's antibodies are involved, and then even some of the work that Brian did in looking at the temperature, salmonella doesn't, at least in Rhinus specifically, Typhimurium to a slightly lesser degree, doesn't like 41 degrees. And it doesn't colonize well at 41 degrees. But little birds don't do a good job of maintaining 41 degrees. And so it hangs out longer, and right around the time the birds are able to thermoregulate on their own, salmonella levels start to tail off. So we've got multiple factors that are in play, sort of keeping the salmonella loose and to figure out how to make salmonella, or make chicken salmonella proof. So, changing gears, still almost all this is, well, I guess maybe not this bubble here, but most of this is all in the gut. So one of the other projects that I've been working on, this goes all the way back to graduate school, is poultry, and specifically working on astrovirus, trying to figure out exactly how it causes disease and what its role is in poultry in general. So we've got some old data about sort of surveillance and looking at different genetic differences amongst the viruses out in the field. I'm not going to talk about any of that right now. I think for the most part, there's a group at Southeast Poultry that's sort of, all they do is sort of molecular epidemiology stuff. So we've been focusing more on sort of the pathological mechanism. And specifically, astroviruses, because there's really no good animal model for astroviruses of any kind. And there's an astrovirus that infects and causes diarrhea in the young of just about everything that walks or crawls. So the question's always been for turkeys specifically, and as astros more generically, how they cause disease. At least in the turkey, and this also seems to be true in most of the other animal models, there's no inflammation. For the most part, when you think of some sort of infectious diarrhea event, there's some sort of tissue damage, there's necrosis, there's apoptosis, there's inflammation, there's something you can point to and say that's the reason why the gut's not absorbing nutrients the way it should. When we give astrovirus infected versus controlled turkey tissues to pathologists, they can't tell them apart. So that's always been the question of, okay, well, if there's nothing microscopically or grossly wrong with the tissue, then why are they getting sick? So a while back, we started working with Anthony Blickschlager, the vet school, to help us with doing using chamber experiments. And we went through and we're actually looking at the tight junctions. So if there's nothing microscopically going on, if we really get down to the ultrastructure, can we see that the tight junctions between the villi are relaxing enough that maybe that's causing any kind of diarrhea? The short answer is we get these really electron-dense regions around the TJs, tight junctions, but there doesn't seem to be any change in what that distance is between those two. But what we do see is sort of a sodium malabsorption. So there's it's not a big change in what's passively fluxing through, but we found, at least, and this is all in the jejunum, I believe, as it turned out, the ileum's a little bit different of a story, but we can get decreased surface expression of NHE3, so you get less sodium absorption, and you get salt imbalance, and so you get sort of an osmotic diarrhea that's happening. And we can go through and sort of see when the birds are infected. You can see actin get depolymerized around the TJs. That's not affecting the TJs, but it's affecting the brush border enzymes and what's being pushed and pulled out of the upper membrane. Other people have described, actually in the 80s, or what's his name, Reynolds at Iowa State, talked about maltose and sucrose activity changing in the guts of a different turkey astrovirus infection. And I think it's sort of probably the same mechanism, that as the cells are picking and choosing what types of transporters and digestive enzymes they put in the upper membrane, as the actin gets screwed up, some stuff gets locked in the membrane, some stuff can't get recycled. And so we think the NHE3 is sort of subject to that. We've been trying to develop the reagents to track that better through during infection. Another part of that too has been almost all viruses you think of causing disease during the replication of whatever it is they do to the cell that disrupts the cell. There are a couple of viruses that, and rotavirus being one of them, one of the non-structural proteins they make while they replicate has toxin-like activity. And in mice models, HIV has a similar non-structural protein that only gets made during replication. We were interested in whether the astrovirus actually had any toxin-like activity, because you anecdotally hear about reports of sometimes some turkey flock supervisors. There's been jokes in our lab that after animal experiments, people a little looser than they were before the animal experiment, and it was always psychosomatic. But if we take the astrovirus and just express the capsid protein in baculovirus, purify it, giving that to turkeys is enough to make them sick. It's not a sustained diarrhea because the virus isn't replicating, it's just sort of one pass, one shot through. But and this is sort of summarizing a paper that came out last year in Bio, where as we increase the dose from 5 micrograms up to 50 micrograms of just the capsid protein alone, we can induce more severe diarrhea. This is sort of one figure to summarize a lot more panels, but again, in the interest of brevity. So we go through, and in these same experiments, we see NHE3 expression's been changed, we see actin's changed to some extent. It's transient, it's a lot harder to find because we're just sort of orally dosing the capsid in there and trying to time when it's going to make it all the way down to the ileum to then sample and look at. So, but this is the first time anybody's described the structural protein of a virus being able to cause diarrhea.
So the capsid protein lies where in the virus?
It's the capsid protein is the coat.
On the coat. Okay.
It is the coat.
Okay.
So yeah, the way this thing's put together, it's one protein that forms an icosahedron by just multimaring itself together with a little protein BB. And so there's, again, I took it out for brevity, but it's got these spike domains. And actually, what didn't make it in the paper, because they wanted us to flesh it out better, and we didn't want to do that yet, if you just cleave the protein so it's just the spike domain, that seems to be all you need. And the spike domain is probably, by all accounts, actually the receptor the virus uses to get into the cell. Whether or not the toxin-like activity is because the spike is binding the receptor, or the toxin activity is some other cell capsid interaction, we don't know. We still don't know what the receptor is. That's another problem. But we can put this in mice. It's really hard to get mice to get diarrhea, but there is some wishy-washy evidence that the mice get diarrhea. More impressively, you can put this on Keiko cells, human colorectal cancer cells in culture, and you can affect their TJs with just the capsid protein alone, the turkey capsid. So again, there's lots of astroviruses out there. So this is not really taken off yet, but it's led to some discussions within the community of, okay, well, if there's, and not to pick on turkeys, but just since that's what I'm working with here, if you had a turkey farm that broke with this, or in some cases a lot of them just had these viruses asymptomatically, because there was an age restriction to it, and they happen to be in, on the Chesapeake Bay, what might that mean for shellfish that could end up concentrating some of the capsid protein that would never infect a human, but if they start eating Orchard Rockefeller, might they get a brief transient diarrhea and no one would ever know because there's no replication going on? And so is there a foodborne disease risk because this is a toxin, not necessarily a viral infection, zoonotically?
I mean, have you looked at the innate immunity associated with this?
Yeah, so that's a whole nother line of investigation where there's no adaptive response that we can detect. In the gut, the infected cells express all the chemokines one would expect infected cells to express to ask for help, but help doesn't show up. And so the gut epithelium itself expresses nitric oxide, and I think the enterocytes themselves end up being what beats the virus back after a couple of days. It's typically self-limiting within 14 to 21 days. And so we've done some experiments, and Rizwana just repeated another experiment a couple weeks ago. When we induce inflammation somewhere else in the body, usually like the abdominal Cephidex inoculation, we get fewer immune cells show up to that if they're astro-infected. And so there seems to be something being expressed during astro-infection that's miswiring or causing the innate system to not respond the way it's supposed to. You can find the same number of cells in circulation, but you don't find them in the tissue where they're being asked to. So,
Have you ever injected this intraperitoneally?
the virus?
No, the capsid.
Capsid, no. Yeah, there's been a slew of things we want to do with the capsid, and that's on the list. But getting enough of it to do those things with
Sure.
has been a challenge. But yeah, no, doing that and as being able to fractionate and sort of let's trips and digest.
How pure was the capsid? Yeah, you express capsid protein in baculovirus, right?
So I got pictures of the gels. It wasn't cut out of a gel, sort of purified,
Okay.
but it was his purified, and then from there, then they actually cleared. So we had a collaborator at St. Jude's do all this because they could afford to make gram amounts of the stuff. So then they would his purify, and then use a secondary column to essentially immobilize the complex and then cleave it off.
Okay.
And then when that would run an STS page and Silverstein, we would see capsid, and in some cases, we'd see a secondary band, which we actually think is the part of the spike that was almost auto-cleaving. But it was pretty clean prep. And there were, not shown here, but there were some initial experiments where we sort of expressed GFP, sort of, or I think it was Laxi instead, and used that as sort of a mock capsid to see if we get if it's just his purified column LU8 was enough to do this or not. And that was no different from the control.
Okay.
Keep trying to bounce through these at Mach 5. So a little bit about genetic resistance. And again, hopefully, I'm not otherwise going to hit this too hard, but hopefully you can sort of see the threads where these things are all interconnected to some extent. So at least in terms of genetic resistance, the one part of the story that where we've worked the most over the last several years is actually looking for this protein activity called MX. So in just about everything else but birds, or at least specifically gallinaceous birds, there's a protein called MX that's thought to be really important in antiviral, specifically anti-flu activity. And this is a little cartoon of how we think there's MXA and MX1, depending on whether you're a mouse or a human. And both of them work in terms of helping to shut down influenza replication in mice and human cells, or most mammal cells in general. And actually, MX gets its name because flu is an orthomyxovirus, and this is myxovirus antiviral protein. And so it's this giant protein that actually, this is one of its monomers. It actually comes together to form these ring structures you can see here that are actually 64 of these monomers all bonded together, and they assemble what this structure here is actually a viral RNA nuclear protein, and they assemble themselves around it, and they've got a GTPase activity that cleaves the viral RNA as a way to sort of inactivate the virus. So people found this years ago in mice, and even to this day, there's only a subset of lab mice that you can ever get to replicate flu. And there's never been a mouse in nature who's ever gotten influenza. The only mice that ever get influenza are the poor bastards who live in labs. And even there's this small subset of strains, and all of them have an MX gene that seems to be inactive. So when people first figured that out, they're like, so this must be the end-all be-all to flu resistance, and this must be why chickens and ducks don't have an active MX, and this must be why they can carry flu, and mice don't. Well, we've got an active MX, and we carry flu. It's a lot more complicated than that. They've got similar structure, so this is out of a paper we did with, Chris and I did with Janet Fulton at Highline. The ribbon structure for human MX compared to the bird MX, fairly similar. There are some moderate differences, but for the most part, it's the same structure.
Where did you get that?
Was that?
Where do you do, you don't do that in your lab.
Mm-hmm.
Do you do it?
Well, so this is protein threading, so we didn't actually do the crystal structure. So
Where do you send that to do the crystal-based structure? Yeah.
So the human MX crystal structure has already been resolved. So there's free programs where you can essentially say, I think this protein is going to look like that protein, and you tell the program, map this onto that and tell me where are there peptides that make it inconceivable for these two things to look like each other. And it'll go through, and it'll take a couple hours, and it'll fold and fold and fold, and it comes back and says, yes, these are a match, or yeah, no, dude, these aren't the same thing.
But there is no analysis. I mean, the computer makes the modeling, but there is no really true analysis.
It's all computational, yeah.
It's not, yeah.
And so it's without the crystal structure itself. This is in silico hand waving, but to a large extent, if there's supposed to be alpha helices here, but then there's no protein, no peptides in that stretch there that would allow for alpha helices, then it's going to come back and say, yeah, that doesn't look the same. And so we can, and what I'm not showing is, so there's MXB in humans has a different structure, and when you thread the two together, thread chicken on MXB, it comes back and says, no, these aren't the same thing. There's some similar motifs, but the structures to them are different enough that says, between the two, this looks more like MXA and not like MXB in humans, or likewise in mice. And so it's not otherwise meant to say that this is absolutely 1000% the way the chicken MX looks, but more along the lines of, okay, do we have the same domains that make, do we have a globular head, do we have a stalk in the middle, do we have a stalk here, and do we have big loops down here, because functionally, this is where the GT-based enzyme activity is supposed to happen. These things are the regions where the monomers come together under van der Waals interactions, and it's thought that these loops, all this is worked out in the human, by the way, it's thought that these loops are actually what confer viral specificity. Some MXs will work great against flu, other variants, even in humans and simian and whatever else, work against other viruses. And it seems like this is actually the recognition sequence. It wraps around the viral RNA and fits in, and once it fits, then this can cut. And so we just want to see, did these look anything like each other? Because, again, working with Highline, they, sort of with them, and they provided us with genetic material, as well as they were doing the sequencing themselves to back it up. We found at least 12 different haplotypes of this gene that no one to date has demonstrated has any actual antiviral activity in birds. And so, at least for me and my cartoon version of how nature works, if a protein doesn't have a function, you get rid of it. You don't make 12 different flavors of it. And so what was especially interesting is Fulton went back through all their deep sequencing going back to 1996. They can actually see different haplotypes changing in certain genetic lines, and this isn't something they're selected for. Which is interesting. You can sort of see, where's my favorite example? 5 is the only one that's in common in both. You sort of see 5 starts off at 0.4 and it sort of trickles along, it ends up somewhere around 0.6. There's a couple here, actually this number 2-1 is actually a new recombination event that only existed in that line. And so they could actually sort of find that these new variants were forming in real time over the decades. In some lines you see certain variants go up, in other lines of high lines you see that same variant actually get lost completely. They went back and did some phenotypic correlation analysis, and some variants correlated with the lines that do really well against Merrick's, but that same allele or that same variant didn't do as well, or was inversely correlated with, egg per day. And to some extent, that tug of war between resistance and production sort of seems like maybe what's in play here as well. But again, so what are all these things doing? So now we're trying to figure out exactly what's happening. In addition to the protein itself, we found lots of variations, not lots of variations, we found about 6 or 7 variations in the 5' untranslated region that actually affects gene expression. So certain mutations will increase or dilute how much gene expression will happen. And we found, even more interesting, lots of variants in the 3' UTR, which nobody ever looks at the 3' UTR. And what doesn't show up, probably in the back of the room, this bold sequences right here is actually the polyadenylation signal. So there's a couple of variants that are missing the polyadenylation signal altogether. And that one, you get less gene expression. Mm-hmm. So there's variants pre and post, as well as variants all throughout the coding structure or coding sequence that are affecting how well it's expressed, and then presumably how well it functions. And so we're in the process now of cloning all of these coding regions and sort of making artificial chimeras between the first part and the back half, and sort of see how they work together. If they're going to form those 60-mer rings, is there something about how they dimerize? That's the reason why they don't work. Can we find which sequence in there might actually be the best, or maybe the best of bad options for birds, but which ones may be the best in terms of antiviral activity, that then Highline can then fold back into their genetic selection and actually start to be one more thing they monitor for, and know which allele they want to keep in the resistant line, or which alleles they should get rid of in the production lines, or the egg per day type lines.
So,
Yep.
so if you're, all right, if you're gonna, develop chimeric molecules, what's your endpoint? And after you have them, like how are you gonna measure the activity?
So there's a couple different ways. One, so we'll focus here where the GTPase activity is. One part of the experiment we set up to sort of figure out how do the different variants perform in terms of GTPase activity. Then another way that people have worked on this with the human is you can do sort of chimeric pull downs where you make one that's got a his tag on it, and then you add your other variants in to figure out how well they dimerize. And then because this thing is such a huge 60-mer, you can actually separate it out of E. coli just by density centrifugation. And we can use that as a way to figure out, okay, well, if we make changes here in the region that's supposed to van der Waals together, how much have we disrupted its ability to form that ring structure?
But the underlying assumption you're making here is that it actually has antiviral activity.
So I guess the way I presented this, yes. But that's not probably really, at this particular point, I don't think there's enough evidence that this thing behaves like MX behaves in other animals. I think the antiviral activity of MX, the specific being able to bind to flu like that, I think that's probably value added. That's something that nature developed later on. This gene is only expressed when interferon's turned on. It's still tightly regulated. I think it's got, and it's this whole complex is actually part of the dynamin family of proteins that are involved in shuttling vesicles around and otherwise clean up the trash and ubiquitination. I think it's part of the sort of generic antiviral response. So when viruses infect, the cell starts to express stuff that degrades RNA, because you don't want any foreign RNA.
Right, so it's an RNA scavenger.
Or RNA or protein, one or the other. It's there to help clean up the mess. And the fact that some of these in mammals or in other species, actually even fish, they've got a demonstration that they work in fish. The only other option, which I think is maybe number 2 on my hypothesis, is we're testing the wrong viruses. So up until about 5 years ago, MXB in humans had no described antiviral activity. 5 years ago, they figured out, yes, it does. It worked against HIV. They were testing against all the wrong viruses. We're testing against the viruses that, from a production standpoint, we care about. I don't know of anybody who's looked at this against Merrick's. They're looking against IBV, NDV, and flu, the viruses that classically that we've been testing against MX in mammals. But flu in chickens is probably something relatively new in terms of, Merrick's has been a problem for fowl a lot longer than avian flu had, well, poultry.
See, but if you look at all the polymorphism that's there, right, it seems to be more of not specific, super specific scavenger.
Yes.
Right, because if they were specific viruses that it was going after, you would expect, I would expect less polymorphism in that sense, because it only would work for, the most lethal, say for example, viruses.
So in most of the humans, where you see the most polymorphisms is in this loop. Everything else through the rest of this is pretty rock solid.
Okay.
Where we see most of our mutation, or in here, you see purifying mutation, so there's evolutionary bias to not change stuff. You see a couple of places under positive selection in the globular head that don't make a whole lot of sense, but nobody's found a globular head in chickens that looks like it's actually got intact GTPase activity. And so this GTPase activity domain is known to be essential for this cleavage to happen. And so either the chicken is trying to otherwise mutate itself back into a GTPase activity that works, or however it works, this isn't important to it anymore. And so, and how we reverse engineer into what did evolution decide to make out of this, I don't know. But it does there's a few papers where it does complex with other proteins in the cell. Most of them are pro-apoptotic transcription factors and things of that nature. So we've done a couple of years ago, we did a couple of yeast-2 hybrid experiments where we fished out a couple of things with the chicken that correlate with the same thing you can yeast-2 hybrid out with the human version. And so again, it's like, maybe it's not functioning like this, it's functioning as a chaperone or some other type of part of a larger complex with other things in the cell. But at this point, no idea.
I have one more,
Okay.
sorry. But, so, is the chicken sequence for MX similar in other bird species?
You mean in terms of activity or
Like,
just
you know,
A's and C's and T's sort of?
yeah, I mean, how homologous are they among, you know,
So between chickens and turkeys, they're like 2 amino acid differences.
chicken and turkey?
Duck is a little bit more divergent, but not a lot.
Not a lot, okay.
And there's no evidence of actual anti-flu activity in either one of those three. A few people have started looking at geese and zebrafinch, but off the top of my head, I can't remember what those papers were. All right, so I'm going to try and kick it in here, but I just sort of summarize, we've, looking at the development of chicken microbiome, how that's related to immunity, how changes in the biome relate to pathogens, which I've got in italics because I don't really think salmonella is a pathogen for birds, but it is for us, so use that word as you see fit. We're looking at how astroviruses cause enteritis and poults, and an immune response that I didn't otherwise show slides, but Jim was nice enough to ask a question when we talk about that. The haplotypes, identified multiple different haplotypes of the antiviral gene in MX, and now sort of working on trying to functionally characterize what those variants actually mean or don't mean for MX function, whatever that is. So just to sort of, those were the sort of the highlights, just sort of to blandly summarize the dossier in terms of publications, so at least, focus on just what's happened here at NC State versus total. 35 peer-reviewed papers, 1 invited, 1 unsolicited review, 4 book chapters, 23 invited symposia, invited seminars, 84 abstracts. In terms of money, 18 externally funded proposals, 13 internally, so in-cars, Ag Foundation, stuff like that. 2 proposals are pending, 33 unfunded proposals. And I did the math last night, and that number does not feel right. It should be 0.48% the way it feels. But I think I front-loaded all the ones that didn't get funded the first 5 years. I've gotten better at picking the writing for what I got better shots at now. So this is based on the number of funded, or number of proposals. Actually went back to the number based on dollars requested, and even that's better than I thought it would be. And then in terms of gifts, tallying up animal health and multi-state project money coming in. Unless anybody's got any questions about those tables. Keep moving in terms of teaching and mentoring. These should go a little faster. So teach 2 sort of primary courses. One is undergraduate agro-security, PO 411. So this is a class I created a whole cloth, and the primary idea here is to get as many kids from across campus as possible. Ideally, I'm trying to work towards having half poultry science, animal science kids, or maybe half cows kids, and half outside of cows. We're going to get folks that are steeped in agriculture, those who come from more business, those who come from more of life sciences, or those who come from humanities, who knows, come together and sort of discuss all the issues, threats, responsibilities, consequences, risks to the food production system. With the, and the sort of pithy line I always use is, you know, at the end of the day, at the end of the class, I want the students to sort of understand how an outbreak of foot and mouth disease in Oklahoma would affect the availability of tube socks in Sarasota, Florida, with just-in-time shipping and governors shutting down shipping lanes, and all this stuff is interconnected. And just because all you know about agriculture is Kroger doesn't otherwise mean that you don't otherwise have to have a part to play in this game. So we have students, you know, have some sort of cursory. I'm not expecting them to be veterinarians or lab diagnosticians, whatever else, but you know, have some sort of understanding about different types of diseases and threats to plants and animals, being able to identify potential consequences of an outbreak or some sort of attack to U.S. agriculture, develop plans. So it's not designed to be a GPA killer, so I have it set up in a way where I put the kids into small teams, which they fuss about every year, because I go through and I specifically mix and match them based on major and what they tell me their interests are, so there's as few people in a team with the same background as possible. And then they have the first part of the semester to build me an attack plan, and they've got to come up with, if it's going to be intentional, they got to identify if it's going to be like terrorism, they got to come up with the geopolitical aims of the group, what they're pissed off about, why they've decided to attack what they've decided to attack, what agent they're going to use, and I tell them if they wanted, and folks have done, you know, act of God or hurricane, or even things as small as farmer across the street is mad at the guy at the other side of the street, so he's trying to sabotage his business. You know, set the scope of what the person's trying to do, and then why have they decided how they're gonna attack, how that's gonna work. They can use, I tell them, they just can't violate Newton's fundamental laws of matter. They can't just say one day they isolated Ebola from their dorm fridge. They've got to otherwise go through how they would get it and have a plausible, even if highly unlikely, still somewhat plausible way that they otherwise this thing unfolds. They have to come up with sort of the biological, ecological, and sort of economic impacts. They turn them in about the middle part of the semester. I grade them, then I scramble them up, and I give each team somebody else's attack plan, and they spend the second half of the semester building a defense. And I tell them they've got to build a defense for the whole thing, but at the end of the day, not everything needs to be SEAL Team 6. If the defense is a $2 lock for the gate, it's a $2 lock for the gate. It doesn't always have to be monitors and cameras and satellites and drones. It can sometimes just be, and you know, we talk about examples from Oregon where there was this cult that was mad about zoning laws, so they tried to poison the town with salmonella at salad bars and like Whole Foods types of cafeterias. Nobody got sick, but they gone around and sprayed salmonella, all kinds of different stuff, because they were mad about zoning laws. Like, you know, in some cases the defense could just be having a better type of grievance thing so people feel like they got heard and they don't otherwise go down that road. So it's been offered 6 to 8 times now. It started off as an experimental course with just a handful of students. It's now up to 40. Len wants to put more in there, and I'm trying to adjust from 5 to 40, and we're trying to figure out ways to take more of it online because it's lost the sort of small classroom conversations. So I'm trying to figure out how best to use forums on Moodle and whatever else to sort of get some of that back, or if it just has to be sort of redesigned from the ground up. So just pull out one part of a quote from one of the student surveys. Found the design of attack and response plans beneficial to the overall learning of the course. I would not change anything about this course, which was nice to see, but stuff's going to get changed. The other classes, so this is a graduate course, comparative immunology. This started off as avian immunology, but there's not enough folks just on our building alone to sort of continue the enrollment there. So I've fleshed it out and got folks from the vet school to come in and co-teach folks who do stuff with zebrafish, cats, dogs, whatever else. We've talked about everything, literally from the immune responses of bacteria, being CRISPR, all the way up to camels and manatees and corals, everything in between. It's taught out of current literature, and so I really stress sort of the critical thinking and tearing the page apart. We do, you know, compare and contrast immune systems of different bird species. I guess I need to change it now because we've been going way down the evolutionary tree. Compare and contrast methodology. So we have the students who come with all different types of model species and what they're working on, and we talk about, you know, well, what may be available for one species, whether it's cattle or mice, that's not available in coral, then how does that change how you design the experiment? How do you still get to do the same thing you want to do? Critical review, immunological research, and then all the exams are these brutal. They get 2 to 3 week take-home tests, and they're all designing an experiment. And often I invent some new species. A couple years ago was fish, bird, pig. And I made them go through, because we talked about how antibody diversification happens differently in fish and birds and in pigs. And somebody finds this missing link, so design me an experiment to figure out which way they do antibody diversification. And so we do stuff like that. The students all complain that the exams are too much work. But then at the end, most of the comments are, I hated how much effort this took, but don't change it. So, what do you do? Other teaching, so helped with 398 a couple times in the past, and now been asked to do it again this spring, focusing, just because I don't have time to come up with new topics, and go with the topic I got asked you last year of antimicrobial resistance. Several years ago, did one on pandemic influenza. I've guest lectured when others have done pandemic influenza in the past. Did a special topics course for some high school kids. I'll talk about that in a couple of slide or two, as well as this protein expression short course. Those two are tied together. Helped develop several short courses. This is one where people pay for on the outside. These two are actually NIH BARDA-funded short courses to teach folks for vaccine manufacturing companies from the developing world how to make flu vaccines. And then guest lectures as needed. Sort of lumping this under teaching, or at least teaching and mentoring. So I ran a NIFA multicultural scholars program that just finished last May. So this had supported 5 students. So we recruited, advised, and managed all 5 of them through at least the 4 years that the grants covered their tuition and stipends. So help them set up undergraduate research experiences, help them get industry internships. These are all during the summer, and then the third summer, between their junior and senior year, did international experience. I'll talk a little bit more of that in a slide or two. 3 of them graduated this past May. 2 of them are off to graduate school. 1 of them is working in a research lab at Wake Forest and trying to decide whether or not she's going to go to professional school or graduate school. And hopefully, I need to talk to them this week for advising, but the other 2 should be graduating this May. We'll find out if that holds true. So this picture here is, the program was written up in the college newsletter because of these 3 went off summer of 16 to Adelaide, Australia. This is them. They spent 3 weeks in Australia doing some sightseeing, but at least 10 to 14 days at a time. They were actually at the ag campus there, sitting in classes, assisting with research, to sort of getting a sense of how things are different in terms of production agriculture. How do research universities support agriculture when you don't have extension? How do they still sort of fill that niche and sort of learn about stuff along those lines? 2 of the students out of the 5 either couldn't go for family reasons or didn't want to go to Australia. It's amazing how hard it is to get away free money. So last May, 2017, the other 2, they went to Northern Ireland and visited with AFBI for 14 days, where again, sort of similar thing. They toured around Northern Ireland, looked at what agriculture looked like there, went to some of the research labs affiliated with AFBI, and there's a picture of them in a stockyard. And so for both of these, I set up a Twitter account for the MSPs as well as an Instagram account so that NIFA could track stuff. So this is cut and pasted out of the Twitter feed for when they were in Australia, and this is the Instagram feed when these 2 were in Northern Ireland. And so actually just a couple of weeks ago, actually early September, maybe more than a couple of weeks ago now, NIFA sent out the announcement for the funding for the next round of MSP grants, and they highlighted us in terms of successful programs in the past of what to emulate. And actually this link here takes you to this story about what their time was in Australia. NIFA followed and liked and retweeted all the posts from from the Twitter feed. And they were over the moon with stuff, and they actually used some of the write-up from the annual report as well as from this in their report to Congress last spring, which I think is why the funding for this went up some. Not specifically us, but they actually were doing a better job this time of collecting data in time or in real time, so using Twitter and using PIs to give them the feedback they needed to go talk to Congress to change the funding stream increase the funding stream some. So last couple of things. So in terms of teaching, this is where teaching and outreach start to get blended together a little bit. So 5-ish years ago, I started this program working with Bertie Early College High School in Bertie, North Carolina. It's a Burroughs Wellcome funded project right now where I'm teaching them how to make recombinant protein in their high school lab. And specifically, they're making recombinant turkey IL-8 for my astrovirus project. Now, we're already making IL-8 for our research anyways. I got other cytokines and things I need to make. The teacher, Bruce Bowler, teacher wanted stuff for his kids to be able to do in the lab. And I said, well, you guys want to make my protein for me? And so we're trying to build it around the idea of a mini biotech company, and if they can ever actually make enough protein that can be successfully used in my lab. Several other folks on campus who work with orphan species said, I got protein I need to make. So hopefully, if we can get this to the point, this could be sort of boilerplate, broader impacts for people writing NSF grants, that instead of buying recombinant protein from Thermo, if you don't need it, turnaround is not going to be quick with these guys, but if you know you're going to need it in a year or 2 years, and they can ever actually get sort of a sustainable system up and going, then your NSF grant, broader impact, we get these kids to make my protein, and off you go. So as part of that, so they were here and they presented about some of the work they've been doing at the Stewards of the Future Innovation Fair back in 2015, and they actually got an honorable mention in terms of best presentation. And then this one I actually haven't been allowed to talk about until the new administration took over because this wasn't open to the public. We actually got invited, this is inside the Eisenhower building, got invited to the White House to talk about, they had a Raising the Profile of Agriculture Summit in DC, and they invited the head of Google's agricultural moonshot program, deans of all the land grant schools, several other movers and shakers. And so this is Matt Lee. He's actually a sophomore now in biology here on campus. This is Cody. She's actually a freshman in this department this year. Emma got in and was going to be a freshman with us, but then family pressure made her go to Chapel Hill, so she's not into us. I think he's at A&T. This is the assistant director assistant to the assistant director of OSTP. And so they were going through and meeting with all these folks, and I would go up afterwards and say, so how was that? Oh, that was really cool. Do you know who that was? No. Well, that's the president of Iowa State. Why didn't you tell me that before? Because you would have crapped your pants. I would have crapped my pants. So they talking to Google, talking to all these folks, and so we actually had them print out business cards. They were handing out business cards to everybody. They had posters they had presented other places, so folks were coming by and asking what they were doing. So then summarize trainees, because I know we're going over now. Visiting scientists, 3 of those, 6 postdocs, 3 PhD students, 3 master's students, 17, been on the committees of 17 other PhD students and 8 other master's students, advised or had 20-some undergrads in the lab, as well as had 1 high school student in the lab, 2 post-baccalaureate students, mentored or mentoring a handful of K-12 teachers, helped advise a couple of MLP students, currently advising or have advised up to 10 poultry science undergraduates, and then served on as external reviewer for dissertation. The last couple of things are really quick. So in terms of outreach, just a couple of highlights. So we've been asked and presented RTP 180 is this lecture series, seminar series, place to get free beer. Every Tuesday or every 3rd Tuesday of the month in the park. They pick different topics. Back in June of 2015, it was ag bio, so it was mostly plants, but I got asked to be the token animal person there to talk about how biotech is changing how agriculture works. So they had Ag Biome was there talking about all their stuff in plants and all the money they just raised from everywhere. And then a couple other folks just talking about the statistics of agriculture in the States. And so I talked about some of our microbiome stuff. Last September, Novozymes asked me to be part of a panel with them at South by Southwest Eco, talking about probiotics and whether or not they can or can't replace antibiotics in production agriculture. And then actually just earlier this month or into September, so I'm on the ASBMB Public Outreach Committee. We participated in a conference at the University of Kentucky, preparing students for careers in STEM, so whether that's academic or industry or whatever else. And a lot of them were interested in outreach, so went and talked about some of the stuff that we've been doing from an outreach perspective. So, and again, I had a hard time in the package sort of trying to decide between what was general public and what was stakeholders and what was these, so I sort of made some arbitrary decisions. But, you know, 9 presentations to stakeholders, 11 or so to the general public, at least one to a group of all K-12 teachers, sort of in terms of a formal setting, and then done a couple of things at local high school, one elementary school and 2 middle schools, one in Eflin and this one's out in Keansville, I think. And then, just because I didn't really seem to fit anywhere else, in terms of outreach, so I've had my own sort of personal Twitter thing for a while, but then I've also, probably for about 5 or 6 years now, I've got one for each of the different classes. So I get students, you know, they can follow the email, but a lot of them still don't otherwise want to check their NC State email address for whatever reason, and that's the only thing that Moodle will push to. So announcements or whatnot for ag security classes or comparative immunology course. Actually, I really use this one as a way to bookmark new papers that are coming out that I don't have time to read. And I can go back the semester before and figure, oh, well, there was a really cool paper on snake immunology 3 months ago. Where was that? So I retweet that out, that I've got to wait, otherwise find it when I need it. This, I sort of try and keep a little bit more firewalled off because I follow a lot of really crazy groups. So Animal First, Earth First, PETA, Mercy for Animals, all of them are here so that in class we talk about some of the different agro-terrorism type things. And there's ALF has their magazine Bite Back. I follow them, and so when they've done crazy stuff, it shows up in the Twitter feed, so then I can make sure that if anything's sort of happening in real time, I can work it into class. I have all the students follow this. I try not to retweet those monkeys, just because I don't otherwise help put their stuff out there anymore. But every so often, there is some stupid stuff that comes, like this one here, that sort of cut off the top I retweeted a couple days ago, was Earth First is mad that Elon Musk wants to give solar panels to Puerto Rico. And why is that capitalist pig going to try and make money off of the poor people of Puerto Rico? He's going to give all that stuff away. It's like, you guys don't like coal either. What do you just can't make some folks happy? So service highlights. This is on the package. I was chair of the science policy committee for FAST before FAST imploded. On the study group for astroviruses for the International Committee of Taxonomy of Viruses, already mentioned the ASBMB, serve on a bunch of different journal boards. Last summer I was co-organizer of the North American Comparative Immunology Workshop. It was actually held here on campus, so it's everybody getting their picture taken about the seal, and this is everybody in niches having a better time than we had at the conference. And then currently IBC chair, have been the radiation safety chair, served on various COWS committees trying to help with current on a research committee, COWS Innovation Efficiency Committee, which Jim was also on, which was not innovative or efficient.
No, it wasn't. It was not.
Cal's Big Idea Task Force, hopefully the dean doesn't see this. Sort of departmentally was involved in helping come up with the building plan for the BSL2, BSL3 thing that's had been somewhat stymied, but now seems like it's gotten a little bit of life again. And it's also because of got sort of looped in with the dean and some others going to University of Adelaide several years ago, trying to develop research collaborations that didn't really seem to go anywhere. Have otherwise parlayed that into, or tried to, and are developing, hopefully next fall will be the first iteration of sort of a poultry-focused undergraduate exchange program. So there's already been an animal science sort of exchange, but it really doesn't work for our students in terms of the classes they would take there, the stuff they would miss here. So we went over, sort of lined up their curriculum, our curriculum, and sort of figured out, okay, what classes do they offer that our students can't get here, and then how do we build a sort of cultural / broader sort of life experience around it, not otherwise having to spend the whole time in a chicken lab. And so I think we've worked some stuff out between the ag campus there and their main campus, and mostly focused around, they have some really cool welfare courses. And we're actually, in one of them, you spend the semester clicker training a chicken. So kids are going to do that, and then get to do some other cultural stuff along the way. And then just sort of, because every time I ask anybody what's actually the sort of metric for full professor, you don't get any kind of cohesive answer other than international reputation, and it's not otherwise sort of crystal clear in the way the packet was together. Maybe it makes sense. To me, I decided to sort of put this map up here in terms of what's in red are countries where I've had visiting scientists or been invited to give presentations, or we just talked about organizing the exchange with University of Adelaide, as well as sending students there a couple years ago for 3 weeks, and so worked out partnerships with them. Visiting scientists from Egypt, invited presentations in France and in the UK, as well as sending some of the MSP students to Northern Ireland, collaborators and invited talks in Canada, Ecuador, visiting scientists from Brazil. So just to otherwise put a finer point on talking to folks outside of the state of North Carolina. And with that, 15 over, apologize, but most of the questions happen in transit, so hopefully there's none left now.
Anybody have any questions? Thank you, Matt. You may
Go for your
take a lunch.
Oh, one, on the grant for the diversity training students, are you going back for that again?
I am not. I mean, I think somebody in the department should, but the amount of time and energy, there's no money for the PI, and there's no money for pretty much anything but the students. That I hate to have to walk away from it, because I think that program built a lot of really goodwill with NIFA. But yeah, I got too many other things